Ever wondered how scientists replicate millions of DNA strands in a short period? The secret lies in a method known as PCR, or polymerase chain reaction. Imagine PCR as a photocopy machine, but instead of documents, it duplicates DNA. So PCR is like a photocopy machine, but for DNA. The first step in PCR is called denaturation. Think of our DNA as a tightly wound zipper. Denaturation is like unzipping this DNA zipper. How do we achieve this? We apply heat, specifically temperatures around 95 degrees Celsius. This heat breaks the hydrogen bonds holding the two strands together. Why this temperature? It's the perfect balance, hot enough to separate the strands but not so hot as to damage them. So, just like unzipping a zipper, denaturation separates the two strands of the DNA molecule. After the DNA has been separated, it's time for the second step, annealing. This is when we lower the temperature and introduce primers. These short DNA fragments are the unsung heroes of PCR. They bind to specific places on our DNA strands, marking the start and end of the section we're interested in. But how do they know where to bind? Well, it's all about base pairing rules. These primers are designed to complement the DNA sequence we're after. So the primers act like bookmarks that mark the beginning and end of the DNA portion we want to copy. The final step in the PCR process is extension. Picture this. DNA polymerase, a remarkable enzyme, takes center stage. It begins attaching nucleotides, the building blocks of DNA, to our primers. It's a meticulous task, much like attaching individual bricks to a foundation. Each nucleotide is carefully selected and added in sequence, extending the DNA strand. This process is repeated millions of times, creating countless copies of the DNA segment. In extension, DNA polymerase acts like a construction worker building a new DNA strand from the primer. Now that we've gone through all the steps, let's summarize the PCR process. We start with denaturation, move to annealing, and finish with extension. This ingenious cycle lets us produce millions of identical DNA strands in mere hours. PCR, in essence, is a simple yet ingenious method to make millions of copies of a specific DNA sequence in just a few hours.